In the third part of the image processing and image analysis workshop, we will start fresh with a new structure. So start up Gedit. And then again, we will browse to the import GeoVol module. So click here on import GeoVol. Click on the browse dialog, on the browse button. And then in your project folder, there's already a raw file prepared for you, which is called AirFilter for Microns. Select this file and click on open. Then you get to the geometry import dialog. And in here, everything is already entered correctly. So you see the domain size and also the correct voxel length because it is saved in the file. Click on image processing to load the file into the 3D image processing dialog. At first, we will turn down the visibility of the segmentation preview a bit. So turn that down up here. And then change the view a bit that we can see a bit more here of the XY plane. Zoom in a bit, and then you can see that there are fibers in there. And these fibers look a bit hollow, but they are not hollow in fact. So they are just a bit brighter in the image at the boundaries and a bit darker in the center, but all this is the same material. If we now re-enable the segmentation overlay, you can directly see that this causes problems in the segmentation. So there's already a lot of noise here in the pore space, but in the fibers, the bright areas are recognized as pore space with a simple threshold, and therefore these fibers are not correctly segmented. Even if you try to use a manual threshold, so if you go over here to global thresholding and select a manual threshold, if you turn it down, you can see as soon as the fibers begin to look better, we get a lot of noise in the pore space. And well, if you turn it the other way around, everything gets worse because the fibers just start falling apart. So this structure is a bit hard for a default thresholding, and usually one would do a lot of image processing here to get to enhance the structure in some way that the fibers get segmented correctly. But this might be a lot of work, and therefore we have another method, we have a great other method which can be used here, and I will show that one to you now. Turn down the visibility to see the structure more, and then we close the global thresholding method here and switch over to the AI segmentation. And with this option, you have the possibility to label the objects in your structure manually. So you can paint in the pore space and can paint on the fibers. And then you train basically a machine learning based on that data and can segment the structure based on that machine learning model. There are several models that you can use. The default is the boosted tree model, which is quite good here and quite fast. But if the case gets more complicated, there are also unit models in here, 2D and 3D options, but these need usually more training data, and therefore also they also need more computational time. So they need too long to compute for this workshop, so we stay with a boosted tree model. Then below, you can select the number of materials. We keep that at the default too, because here we have the pore space and the fibers. And then we can select the materials. We keep them as they are here. And then you have down here the selectors, either the mouse controls, then you can paint or label something, or you can delete labels. So we start with the pore space, and then we switch to the paint mode, and you can see now the white circle here, and now we have basically a paintbrush and can paint in the pore space. So let's label some pore space. You don't see anything right now. That's because of the visibility slider, which is still turned down. So if it's also the case for you, turn up the visibility slider, and then you start to see what you have painted. And you can also see that the overlay switched to AI labels. Now we have marked some of the pore space, and we clearly also want to label some of the fibers. So we switch the material here. Then you see that the circle turns red, but we can directly see that our paintbrush here is a lot too large for the fibers. So we turn this down here, for example, to 4 voxels. Let's see, no, that's still too large. 3 or even 2 voxels might fit in one of the fibers. So if we select one of the fibers here and paint in there, and for example also down here. And what you have to make sure is that you label the boundary, but only if you're really sure that this point is boundary and does not belong to the pore space. So we zoom in up here. Zooming in now works with the holding the control key and the mouse wheel, because the right mouse button is now used by the painting modes, and you can see that it's a bit difficult here. So we should delete a bit of the labels here. So we switch to the delete mode here, and then you can delete the parts here at the boundary. You don't need to be too accurate, but it's always bad if there are wrong labels, because they might create wrong label data in the end. So I'll also remove this part there. I think this looks better now. Switch to the mouse mode again, zoom out. Then you see I've labeled some of the pore space, some of the fibers, and based on this we can train the model. You do this by clicking the train button here at the side. Click on train, wait until everything is computed, and then we have a trained model. And based on this model we can create a preview how good the segmentation looks on our structure. So I click on preview, and we can directly see that it already looks quite good here. So we have a look in here. The fibers are nicely segmented, and you can also switch the overlay up here to you can also switch the overlay to the outlines, so you can directly see the boundaries between the fibers and the pore space. In case that you're not really happy with the segmentation already, you can switch back here to AI labels and change the model again. So you can just add more labels. For example, you can add more labels, for example, also in the other directions. So for example, I switch here to the X direction and paint some of the pore space in here, and you can also paint the fibers in this direction. So for example, those here. And then you can continue training, continue previewing until you're happy. If you're happy with your model, you could either directly segment, but you can also save the labels for later. This is via the Save Label button here. So you can save the labels. There's also already one example file here in the folder. Type in a file name and load the labels later. Or you can, if you have trained, you can also save the complete model and load the model for another structure. So you could train the model on one structure and reuse it on another structure. I will quickly load some labels here because I prepared some labels for this workshop, which you can use too, so that we all have the same structure later, or just in case if your segmentation doesn't look very good later, you just can load these labels and everything will be fine. So click on load labels, and in your project folder there is a file, AirFilter training data. Select that, click on open, then click again on train because it's only the labels and not the model. And we can again have a look at the preview. Well, it still looks nice, and based on this we can create the segmentation, but before we will change the result file name to AirFilter 4microns segmentation. So we change the name, click on create segmentation. Then we wait until all the slices are segmented. And then again, you get the result file. In the result viewer, we see the method of the segmentation, everything that was done, and again with all the information that you need to reproduce the segmentation here. Now we can close this dialog, and we can also close the image processing dialog because we're finished with the segmentation. Now again, we are here back in the Geodict main user interface, and we can again see here the volume field and the segmented structure. You can again toggle the structure on and off, and also the volume field on and off, as before too. And again, you can also slice through the structure and see the segmented structure here. Let's switch to the 3D view and directly disable the visibility of the volume field because we only see black here. So disable it, and then you can have a first look at the segmented structure. To see the outlines a bit better, you can go over here to the camera tab and enable show outlines and show halo. This enhances a bit the contrast here, and then we can see that the segmentation of the fibers already looks quite nice. And now we want to know a bit more about the fibers in this structure, and for this we switch to the FiberFind module, which helps us to identify the fibers in the structure. So go up here to Analyze FiberFind, and then we have several options. We will use the we will have a look here at the Estimate Fiber Orientations option. So click on this option and then the Edit button. And again, we change the volume, the result file name to a name that contains the structure name. It's always easier to find results later on. So call it AirFilter 4microns Fiber Orientations. And then we have several options here. We keep the star length distribution option, and we check to write a grain orientation file here by writing a GOF file. And since we're not so much interested right now in the orientation tendrils, you can also enter a 1 in here, and we want to have a closer look here at the grain orientation file. So click OK and click Run. Then the computation will take some seconds. And then we get again the result file. And here in the report we don't see much because we only selected one orientation tensor. But as I said, I think it's more interesting to have a look at the fiber orientation visualization. And therefore we switch to the tab with this name here and select the geodict orientation file and click on load GOF file. We can keep all the fields checked in here, but if you only want one of them, you can also check only one of them, but I'll keep all checked. Click OK. Wait until the fields are loaded. And then we can close the result file. And now we see the structure and the volume field in here. First, let's disable the structure in here. And there's not much to see yet. That's the case because the volume fields are selected to be visible on all the material IDs, and we don't want to see the orientation in the pore space because there are no fibers, so disable this here. Then you see the fibers, and then we also switch to a more interesting field than the default field here, and we select the angle to the X direction. Make sure to select this also under the threshold by option up here. And then you can see the computed directions of the fibers. Depending on the X direction, you can see what is marked in blue has a very small angle to the X direction, and the more you get closer to red, the larger the angle differs from the X direction. And then here with the threshold by option, you can threshold the angles starting at -pi here and clip away the fibers the more they differ from the X direction. In the next step, we not only want to have a look at the orientations, but we want to identify single fibers in our structure, and usually this is not very easy because everything hangs somehow together here, so the fibers overlap each other, and there's no clear and easy way to distinguish them from each other. Therefore, we will use the FiberFind AI option to label the individual fibers in the structure. This module uses a machine learning model which was trained on a lot of geodict structures, and this machine learning is then used to identify the fibers. There come already pre-trained models with geodict or with FiberFind AI, and you can directly use these models on your structure. We will do this in this case here. So select Identify Fibers AI. Click the Edit button to open up the fiber identification options. In the beginning, it might take some time until the window opens, because at the first start, geodict has to look if a proper GPU or a proper graphics card is in your computer, and this testing just takes some time. In my case, I opened the window already before, so therefore we didn't need to wait here. You can directly see it here under the solver options that a graphics card was found, so you can check this out. If no graphics card was found, it can also run on the CPU, so on the central processing unit, but this will take much longer. So if you only have a CPU and not a GPU, it's probably better if you don't start the analysis and load the results then from the file that we delivered. So go here again under the fiber identification tab. I will shortly explain the options, but we don't need to change anything, so we keep the machine learning option. We want to use the current structure because well, that's a structure we want to analyze. We can also keep the default network, which labels the center line here. We want to analyze all the materials. Below here we can select a threshold which decides which voxel belongs to a fiber center line and which belongs to the pore space. Basically, FiberFaint AI computes a field with probabilities for each voxel, if this voxel belongs to pore space or to the fiber. And a probability of 0 means that it belongs to the pore space. A probability of 1 means it belongs clearly to a fiber. And for everything in between there are different probabilities. So, and since 0.5 then means 50% probability, it makes sense to divide the field here at 50%. If the model doesn't fit very good to your structure, it might be necessary to change the threshold here, but usually we can keep it at the default. Up here you have an option similar to GrainFind to remove fibers at the domain boundary. We will keep this unchecked here because all our fibers, or nearly all our fibers, will touch the boundary, and this would remove everything, so this doesn't make sense. And here finally we can fit fiber shapes into the structure, which will create geodict objects, which we also can edit later. And it makes sense in this case here to use curved fibers. Finally, under the output options, make sure to check Save Fiber Orientation Distribution as GOF, as grain orientation file. We can then compare that later to the grain orientation file we already saw. So click OK, and then click on Run to start the computation. This computation may take longer or shorter depending on your graphics card. So if you have a newer NVIDIA graphics card, it will be faster, and it will be probably also faster with a not so new card than on my computer, because my graphics card also does the video recording, which also takes resources. But if you have an older card or are only running on the CPU, it will clearly take longer than here. So if you want to see a bit more what it's doing, you can also open up here with the two arrows the log, and you can look how the algorithm is proceeding. In the different windows it investigates in the structure. Now it already saved the first file, and then it starts to do some post-processing, so to combine or to compute the individual fibers based on the labeled center lines in the structure. And now also the orientation file is computed. So now, as always, a result file is opened, and in here you get directly a report, as also in the other cases. And in this case here, we directly see that 341 fibers were found in the structure. You get information about the orientation tensor, and there's a lot of data, a lot of more data. I think we won't look into detail in here because that's a lot of information, but we want something to see, so therefore we switch to the fiber identification visualization tab here. And first we want to look at the identified fibers, so choose that one and click on load G32 and click OK to load the index image. We want to set the structure to invisible because we want to see the grain index image. Close the window And now you see the labeled fibers in here. I think this directly looks quite nice. You can see the individual fibers colored in different colors, and in most cases the network works quite well. I think here's one case where we see where two fibers are not separated properly, but in other cases, even at the boundary, the result looks quite well. Even in this case where two fibers are running in parallel all the time, they're quite well separated. Only here are some slight mistakes, but these are also boundary effects. So if you switch to the boundary to do the volume field and clip a bit in the Z direction, you can clip in here and see that the segmentation looks quite nice. Let's switch back to the result viewer. Oh, I closed it, so we have reopen it again. So open here the identify fibers result. And then there is a lot of other options we can look in here. So you can, for example, load a fiber fit, which might be quite interesting to work further with the fibers. So we load this the GAD file now. Set the structure to visible again. And then I uncheck the volume field. And at first glance, this looks quite, looks nearly the same as before, but this is not the original structure, but a structure made of geodict objects. And you can see this here at the green dot at GAD objects. And if you right click on it, you can even edit the objects in there. So a dialog options which with all the fibers. So here the 341 fibers. For each fiber you can then change parameters. So you could take one fiber and change the diameter, for example, or change the position of some of its points. So there's a lot of options that you can do here. Back to the result viewer. Then what also might be quite interesting is to have a look at the labeled structure, because that's the basis that was computed before. And there you see really the output of the neural network, that it finds the center lines of the fibers. So if we go into the visibility options up here and uncheck ID1, you can only see the center lines that were labeled for the structure. So this might also be interesting to get an idea how the algorithm works behind here. Switch back to visible, close that again, have another look at the results. What might also be interesting is to have a look at the grain orientation file, or not grain orientation file, geodict orientation file. I often confuse the G here, but we don't have grains, we have fibers. So there we're back in the viewport here, and again we can disable the visibility in the pore space for the volume field and disable the structure, and then you see again an orientation file, but this file looks a bit smooth, or a lot smoother than the one before, the file which was computed here with estimate fiber orientations was a lot more noisy, but this one here is now based on the identified fibers, and therefore it's not that noisy. And even if the threshold through here, you see that there are individual pieces of the fibers which are labeled to one orientation. Now there would be still a lot to see here in the results. For example, there is a lot of new post-processing options where you can threshold fibers based on properties, for example, But since we are running a bit out of time, I can just give you a hint there that's quite useful and interesting to have a look at the post-processing options in here. But I would say that we finished the workshop for now. You can now again ask questions, and many thanks for being in the workshop with us.